Awesome. Thanks for the introduction. Hey everyone, I'm Mark. I'm not going to talk about kind of how I got here because that was covered quite nicely in the introduction. But I want to dive straight into the topic. I'm going to talk about quantum classical hybrid algorithms for protein folding and docking. So that's mainly the R&D aspect of our company. And I'm going to start slowly and give you kind of a quick introduction to quantum computing, high-level conversation about quantum algorithms, and then I'm going to dive into 3 specific applications that we looked at. And I'm going to try to cover them from kind of a high-level perspective to just kind of get you into the mindset of how to think about framing problems in the quantum computing world and how to actually kind of go through the workflow in order to implement and run these types of algorithms. So first of all, quickly about our company, we're focused on pharmaceutical drug discovery. And when you think about drug discovery, you can think of it as a key and lock principle, where the lock is a protein target, like this one here, that is linked to a certain disease in your body. So that might be a protein that is too active, too upregulated. And in order to cure the disease, pharma companies are trying to find molecules that bind to the binding pocket of that protein, thereby, for example, downregulating it and curing the disease. And pharma companies traditionally have done that in the wet lab. They just synthesize millions and billions of compounds, screen them against those proteins, and then do a local search and local optimization around the molecules that seem promising. And what we're going to do at ProteinQure, what we're already applying, is that we model these things in silico on a computer, on supercomputers, in order to kind of iterate through many, many molecules and trying to converge on good binders that you then can sell to pharma companies and collect milestone payments on. So it's important to kind of be aware of the landscape of drugs. Usually, historically, pharma companies have mostly focused on what's called small molecule drugs. You can think of it as aspirin or ibuprofen, small chemically synthesized molecules. They're usually in the range of tens to hundreds of atoms. Then on the other end of the spectrum are antibodies, which are large proteins. They tend to have 10,000s of atoms. They're huge molecules, and they tend to be able to cure diseases like rheumatoid arthritis or neurodegenerative diseases. But they're also very expensive to produce. And the field that we're playing in is kind of this in-between range, medium-sized proteins, peptides, that you can synthesize and that have very unique capabilities to curing diseases that we currently don't have drugs for. And the reason why they can do that is because our bodies know how to handle proteins. Our bodies know how to break them down, which means they tend to be less toxic. And because they're much larger, you can basically inject them into your body, and they really only do that one thing that they're meant to do, bind to that one single protein and not bind to all kinds of other proteins, which these guys like to do, which causes side effects. And so really there's a lack of tools, a lack of computational techniques to design these types of compounds because the search space is generally too big. And that's what I'm trying to kind of illustrate here. So if you consider a protein of length 50 amino acid, so amino acids are the fundamental building blocks of proteins. There's 20 natural amino acids. If you consider all the possible sequences that you can create for a protein of length 50, you get 10 to the 65 possible sequences. The problem is that right now we only know 150,000 protein structures. They're publicly available in a data bank. And that's obviously a tiny amount compared to this massive state space. And even if you would know all the protein structures and all the protein sequences that occur in nature, you would only cover 10 to the 15 molecules. And so the issue and the question that we try to answer at ProteinQure is how can we develop techniques that help us venture into this unknown space and not just train a machine learning algorithm on this tiny amount of data and trying to kind of stay close to known structures, but really trying to develop new scaffolds that you can work off and make drugs out of. And so the technology at the heart of the company are 3 technologies that we think are synergistic. On the one hand, there's molecular dynamics simulations on classical supercomputers that model the protein folding and docking component. I'm going to get back to that on the next slide. The problem with those simulations is that they tend to be very slow. And that's where quantum computers come in. We're already developing algorithms today that will help us eventually, when these computers are large enough, to scale these simulations and do them faster such that we can iterate quicker and try out more molecules. And then lastly, we are doing R&D when it comes to reinforcement learning because it provides quite a nice approach to learning without a lot of training data. You can operate in low data environments because you basically have your agent kind of learn like a dog, and it just tries out, and it can interact with a simulation environment, which is modeled by the MD simulations. And so putting this together basically ends up in this loop where you start out, at this point, you start out with a random amino acid sequence, or ideally maybe with a good initial guess. And the first step is protein folding. You're trying now from that two-dimensional sequence of lattice, predicting a three-dimensional structure of that protein. And it's important to know that like the drugs that we consider are proteins themselves, the protein target, it's also a protein, kind of gets confusing sometimes, is provided to us by the pharma company. They usually have spent a lot of basic science in the lab trying to figure out these structures. And we can then, in simulation, compute the binding score, which means how well does that key fit into the lock. And then depending on that outcome, we can reward or punish a reinforcement learning algorithm, which in turn changes the amino acid sequence. And then we keep iterating through the cycle until we converge on a drug candidate that we can actually sell to the pharma companies. So it's important to point out that we do not sell any software. All the software stays in-house. We're purely like a molecules as a service company. It's actually a new thing that has been popping up quite a lot with startup companies. And it's something that pharma seems to be very comfortable with doing because they have a problem early on in the pipeline sourcing a lot of initial hits, which they then can further develop. So now I want to quickly dive into the basics of quantum computing for the people that haven't attended Colin's talk about like the D-Wave's approach to quantum computing. So the idea here is that a classical computer is based on bits, which are either 0 or 1, whereas a quantum computer is based on this fundamental unit of a quantum bit, short qubit, and you get a much richer set of states. You can basically be in these superpositions where you can have 50% 0 and 50% 1, and you can really change these percentages as you want. And by doing so and combining many qubits with each other, you can essentially get an exponential increase in the amount of memory that you temporarily have in that quantum computer. So a classical computer with n bits can store n bits of information, but a quantum computer with n qubits can effectively store 2 to the n classical bits worth of information. And to make this a little bit less abstract, how do you actually implement a qubit physically? There's a relatively simple example. When you look at a hydrogen atom, it has a single proton at its core and an electron surrounding it. And usually that electron finds itself in the ground state, which you can define as the 0 state. And in order to do a bit flip operation, you can send a laser with exactly the right amount of energy to that electron, which makes it move up into its first excited state, which you can define as a 1. And you can create these sort of superpositions by shooting a laser that has not enough energy for it to completely transition into the excited state, which means it has a little bit too much energy to stay in the ground state, but also not quite enough to fully transition. And that's how you can get these superpositions. And you can effectively take any two-level quantum system and turn it into a qubit. And all of the different quantum hardware providers are taking different approaches of building them. So then just briefly, a lot of popular news, if you read them, people always talk about the number of qubits. That's kind of like the single metric that everyone is focused on. And it's something that is quite important to realize that it's not the most important metric. It's really a combination of the number of qubits, but also the quality of your qubits. Because there's a lot of errors in these devices nowadays. And it's really important to keep these errors low while still having a large number of qubits, as well as the connectivity on the chip. These qubits are not actually all-to-all connected, which means you're restricted in terms of the algorithms or the kind of layout of algorithms you can run on them. And that's why, for example, IBM has introduced another metric, because people tend to think in single numbers. It's something that they understand easier. It's basically a trade-off, this quantum volume of error rate and the number of qubits. And you're really seeing here that just increasing the number of qubits while keeping the error constant doesn't really increase your quantum volume, whereas you really try to optimize both of those metrics at the same time. So I also want to briefly talk about this notion of quantum supremacy. This is mainly the reason why I'm here today, why the field of quantum computing has been flourishing in the last couple of years, because people expect this tipping point where you have a quantum computer large enough such that it can actually do a computation faster or better in some way than a classical computer. And that's something that people are expecting to happen sometime this year or potentially next year. But also here, I want to point out that there's different notions of quantum supremacy. So what a lot of people kind of expect in the the first demonstration is going to be this idea of weak quantum supremacy, where you essentially solve a very esoteric mathematical problem faster on a quantum computer than on a classical computer. But it might not translate to business value or like running on actual data and have some sort of meaningful results for the business world. And that's really why we're aiming for this quantum advantage, which really means solving valuable problems faster than a classical computer can. And before diving into the specific algorithms, I also just kind of like level the expectations of what types of algorithms I'm going to talk about. A lot of people know Shor's algorithm, for example, which kind of threatens RSA encryption, and certain other popular quantum algorithms that seem to have exponential speedups. But I call them kind of old school quantum algorithms right now because they tend to be very wasteful when it comes to the resources. They need error-corrected qubits, which means that you roughly need like 10,000, if not 100,000 of physical qubits in your device. And that's something that's probably going to take like 7 to 10 years to really come to full fruition. Whereas there's a new approach, which is called variational quantum algorithms or hybrid algorithms. And I stole that picture from a quantum hardware company called Rigetti, and it kind of nicely shows that the idea is to have a quantum processor, which they call QPU, like run hand in hand with a CPU and kind of work together, and both of them kind of leveraging whatever they're good at. And so the idea really is to run the algorithm mostly on the classical computer, but then only outsource some of the heavy lifting to the quantum device. And so if you put these things on a spectrum in terms of the speed up that you can expect from these types of algorithms, kind of today at this quantum-inspired stage where people basically implement algorithms that derive some sort of insight from the way quantum computers work. Then we have these variational algorithms, which people expect to be useful in the next 2 to 3 years, and then the full-scale quantum algorithms kind of 7+ years out. And today's focus of the talk is mainly going to be on this variational hybrid world of quantum algorithms and how you can apply that to the life sciences. So the very first paper that we wrote as part of the company, when we're still in that Creative Destruction Lab incubator program, was we wanted to look at protein folding on quantum annealers, which are the type of quantum computers that D-Wave is building. And whenever you try to run something on an actual QPU these days, you need to somehow coarse-grain and like slightly simplify your problem because it's going to be a proof of concept. So in our world, we have a fully atomistic protein model, which is too complex at this point. And what we do is we coarse-grain it by saying, OK, we're going to bundle up a bunch of atoms into a single amino acid unit, and we're going to represent the protein as beads on a string, where each bead represents an amino acid. And also, instead of folding it in continuous 3D space, we're discretizing the space that this protein lives in. And we essentially took like different types of lattices. In this case, that's a cubic lattice. And the problem of protein folding then boils down to finding self-avoiding walks on that lattice, which minimize the energy. And so to zoom out quickly and talk about a workflow, a typical workflow that you go through when trying to implement a problem on a quantum annealer. You start out having a problem definition. For example, you want to solve the traveling salesman problem, or in our case, we want to fold a protein. Then the next step is you need to encode your problem in what is called an Ising type Hamiltonian. It's a quadratic equation, as you can see up there. And there's a lot of ways of actually mapping problems to this type of equation. And what you see is that there's a quadratic term which has a coupling, Jij, which is a coupling between 2 qubits in the hardware, and a bias term, the Hj, which is a bias on a single qubit. And once you formulated your problem, basically those couplings and biases are knobs and parameters that you can tune in the hardware. So that's how you kind of load your problem onto the chip. But before being able to do that, you need to do a minor graph embedding. Because the problem is that in this step, there might be in this quadratic term, qubit 0 and qubit 100. They need to interact with each other, but they might not be connected on the hardware itself. So that's where this minor graph embedding comes in. Basically helps you mapping your problem onto the actual chip architecture. And then once that's done, you can either run it on the quantum annealer if you have access to it, or you can run it on with a classical solver and simulate it. So really what we're in now is like, how do we encode this problem into an Ising type Hamiltonian? And there was some early work from a group in Harvard. And what they did back in the day is they took a 2D lattice and folded a protein with 4 amino acids on that grid. And what we did is we generalized that approach to cubic lattices, 3 dimensions, where we basically encoded the directions on the lattice with 3 bits each. So basically, the structure of a protein is fully determined by the turns that you take starting from the initial amino acid. And we didn't just like generalize it to 3D, but we also improved the implementation in terms of scaling, which was necessary in order to actually kind of push the amount of protein length that we could do on the hardware. And so we actually implemented that on the D-Wave 2000Q, which is the current chip that is online that has 2,000 qubits. And what we did is we did 8 amino acids on a 3D lattice and 10 amino acids on a 2D lattice. And you can see those final structures, and what the red dotted lines are, they kind of show the interactions that you see on the lattice, like which amino acids are neighbors and are basically like realizing a part of this potential. And what I want to point out here is that, for example, this DA interaction, you can see that in the experimentally solved structure here as part of that helix component of the protein, as well as, for example, that KQ element, which is over here. So really, even though we went into a coarse-grained world, you can still derive some sort of high-level information from coarse-grained models. And that's kind of the idea that is part of this first step where we try to predict the three-dimensional structure from a two-dimensional amino acid sequence. And there's a thing called restraint-based molecular dynamic simulations. So you can think of a molecular dynamic simulation as starting from an extended state. It's kind of like a long string of that protein, and it's going to spend hours, if not days, just trying to kind of curl up the ends and slowly converging to some sort of globular structure. And that wastes a lot of time, resources, and money. And so the idea with restraint-based simulations is that you can take external information that might be those contacts from a lattice fold, it might be evolutionary data, it might be low-resolution NMR or cryo-EM structures, and you can use that information to already kind of like pull your simulation in the right direction, which saves a lot of time because you don't waste all this compute time just trying to like fold it up in some sort of way. And that's something that people have done in traditional computational biology quite a lot, and it shows that there's a constant improvement in terms of the runtime, which doesn't tend to excite like theoretical computer scientists, but it definitely excites us because it translates to a lot of savings. So this idea kind of like drives a lot of the components that I'm going to be talking about from now on. The next step was we wanted to try to benchmark other hardware providers and see how far can we push their hardware, because they have completely different frameworks, different implementations. So we work with Rigetti and IBM on this universal type of a quantum computer, which means it's much more like you can basically have a set of logic gate like a classical computer, just that they're quantum gates, and you can arrange them in any sort of way, computing any function to any error or like approximation that you want. And the workflow here looks similar. You have a problem definition, but instead of having to map it to an Ising type Hamiltonian, at this point you can pick a quantum algorithm, and there's really like a whole zoo of algorithms already out there, and you then have to write down a quantum circuit that basically represents that algorithm. And it's really still quite manual, this kind of work. Like people starting to build abstraction layers, but at the end of the day, you often still have to actually look at the actual circuits. And then because that again might require certain qubits to interact with each other that are not neighbors on the chip, you need to compile it using a quantum compiler, and then you can run it on the quantum processor or on a simulator. And so what I quite like is kind of contrasting those two frameworks and kind of showing the similarity where in the universal world you can pick this quantum algorithm and formulate it as a quantum circuit, and basically that's encapsulated in mapping your problem to an Ising type Hamiltonian. The algorithm was chosen for you because quantum annealing is already kind of, it's a special purpose device, and then mapping it to this Hamiltonian is basically compiling this quantum circuit, even though it's not a circuit. And then the compiling step is equivalent to the minor graph embedding because you're trying to take your problem and map it onto the quantum chip. So for this universal quantum computing world, we picked our quantum algorithm to be the QAOA, which stands for Quantum Approximate Optimization Algorithm. Some people call it QAOA. And the way it works is you have a quantum circuit that has some gates that are parameterized. And at the beginning, you just pick those randomly, those parameters. You run your quantum computation, you measure it at the end, and you feed the output into a classical optimization algorithm that tries to optimize those parameters. The new set of parameters is programmed into the quantum device. You run it again, and you keep iterating, keep optimizing those parameters in your circuits. It's kind of like training a neural network. And the hope is that you can actually, the problem that you encoded, you can actually get very close to an approximate solution. And I want to highlight also that it's very similar to what quantum annealing is actually doing. So the way quantum annealing works is you start from a simple initial Hamiltonian that describes your processor, and then you slowly evolve it into your problem Hamiltonian, which is basically this equation that describes your problem, and then you freeze you measure, and you hope that the result that you read out corresponds to a low-energy solution to your problem. And what the QAOA algorithm is doing, it's basically doing what they call a trotterization. It's kind of like a step function, a digitized way of doing this. And if you have a QAOA depth of 1, which you can think of as like layers in a neural network, you can have like different depth for those types of algorithms. So QAOA depth 1 looks like this, and the more you increase this QAOA depth, the more you approximate the smooth trajectory that you would get from a quantum annealer. And you can prove that in the limit to infinity, it actually perfectly equals each other. So we again encoded the problem, or like basically we had to somehow frame the problem in a binary way. And this time we chose a different encoding. So we actually one-hot encoded every direction that you can take on that cubic lattice, which is a little bit more wasteful when it comes to the number of qubits you need. But we wanted to showcase a particular example. We didn't just want to replicate what we did on the quantum annealer, but we wanted to show that there's something different that you can do on the universal, which you couldn't do on the quantum annealer. So it's going to get a little bit involved right now, but like I'm trying to make a point why we did this one-hot encoding. So there's a notion of Hamming weight, which is essentially the number of ones in your bit string. So this bit string has a Hamming weight of 0. This bit string has a single 1, so it has a Hamming weight of 1, and this bit string has a Hamming weight of 3. OK, so why am I telling this? Because if you have a protein that has to take 2 turns on your lattice, and you get this solution, you're going to be wondering, OK, this actually means like the turn went to the right, but what does this 6-tuplet of bits stand for? It doesn't correspond to any direction on the lattice, so it's an invalid solution. Same with this one. This 6-tuplet of bits doesn't encode anything. It doesn't mean anything. So it's also not a valid solution. So we can say that every solution, or for a solution to be valid, it needs to fulfill the condition that every 6-tuplet needs to have a Hamming weight of 1. And this realization kind of leads to this idea, where you have the entire state space of possible bit strings, possible solutions that you could get out of your quantum device. But we know that, for example, this string doesn't have any meaning because it doesn't encode any directions. Same with this one. So really, this space is useless for us. We know that we're not going to find any solutions there. And we also know that there is a feasible subspace within that larger state space that corresponds to all the feasible bit strings. And that's called the feasible subspace, where really every bit string has, like every 6-tuplet has a Hamming weight of 1, and we know that these are all valid solutions. They might not be the optimal solution, but they're valid solutions. And so the idea here is, can we make an algorithm, can we do this QAOA algorithm such that it stays in here instead of searching in there? And the answer is, yes, we can. I'm not going to dive into like the mathematical details, but basically there is a component in that algorithm which is called the mixer Hamiltonian, which kind of shuffles around the values of the qubits. And that's something that is hard-coded in the D-Wave machine. It's basically dependent on the hardware implementation. Whereas on the universal quantum computer, you can tweak that mixer Hamiltonian however you like. And we tweaked it in such a way that it would, like if it shakes and mixes the qubits, it would always make sure that it doesn't leave that feasible subspace. And so we ran simulations on this approach, where on this side you can see different types of mixer Hamiltonians, this shaker Hamiltonian basically. And if you just run vanilla QAOA without our new approach, you get 10 to 15% ground state probability, measuring the correct solution. If you do the approach that we're proposing, where you use these fancy mixer Hamiltonians, you can get up to 30 to 40% ground state probability, which becomes really important if you're trying to solve big problems. The higher that ground state probability, the more likely is it that you find the optimal solution. And we then basically like also estimated the gate depth, like how many gates do you need in order to implement this algorithm? And that's really the main issue here. Like if you look at the total depth on the 19-qubit device by Rigetti, we need roughly like 800 to 900 quantum gates. And actually like the C0 count, that's like a 2-qubit gate, you need like roughly 350. And the problem is that Rigetti can only do 10 right now. So that's why we had to basically, like those were on larger molecules. So we basically had to restrict ourselves to 4 amino acids. We managed to fold that on the QPU, but we couldn't push it to larger quantities because simply that number is way too large. The hardware needs to improve. Because basically after doing 10 operations, you might as well flip a coin. You just get random noise. So yeah, that's really the issue here. So that's the protein folding component. Now I want to dive a little bit into molecular docking. And we work together with Xanadu on this. Xanadu is another company based in Toronto. They do a photonic quantum chip. They're still building that. They're looking to release it by the end of this year. So that means they're using the particles of light to do the quantum computation, which is nice because D-Wave, IBM, Rigetti, everyone needs to cool their quantum computers down to almost absolute zero. Whereas Xanadu, if they manage to build this processor, they could do it at room temperature. And it's basically just light flowing through an optical array doing the quantum computation as it goes. So we looked at molecular docking. I just want to bring up this slide again where you have a molecule, you have this protein target, and you're trying to figure out how well does that key fit into the lock. And that's the docking problem. You have a lot of possible configurations, and you're trying to find the lowest energy one. And so what we did, we simplified the problem, as always. Instead of taking an entire molecule, which would be too complex, we reduce it to its pharmacophores, which is basically just a fancy name for chemical groups, important chemical groups. So for example, you have some hydrophobic groups, you have some aromatic rings, hydrogen acceptors, hydrogen donors, and you simply mark those elements, those groups, you label them as a single point with coordinates, and that's how you can now represent your molecule. It's something that pharma companies have been doing for years because it also speeds up the classical algorithms to search through databases and stuff. And to kind of get to the next step, I just want to briefly talk about what is a graph. A graph is simply a set of vertices connected with a set of edges between them. And there's a lot of ways of formulating any kind of problem as graph problems, and there's also very good heuristics in the classical world to solve problems relating to graphs. And also, graphs tend to be a very nice breeding ground for good applications for near-term quantum computers. A lot of these algorithms are very good at solving those. And that's why we basically took the molecule, reduced to its pharmacophores, and we can just represent it as a graph. You basically just represent every pharmacophore as a vertex, and you draw edges between them. And we then went a step further and said, okay, well, you can now label those vertices. So A stands for aromatic ring, HA stands for hydrogen acceptor, and so on. And you just draw an edge between each of those and measure the distance in the actual molecule. And that's how you come up with this graph. And so then the next step is you do this for your protein target, which is the receptor, and the ligand, which is the drug molecule. You both represent them as these pharmacophore graphs. And you then create a hypergraph. I'm not going to dive into the specifics of that because it gets a little bit too involved. But basically, you can combine them into a joined graph, which tends to be very large. And what that graph is trying to do, it takes these two graphs and it tries to superimpose them in many different ways, trying to apply a potential function and trying to find out what is the lowest energy configuration of these two graphs. And then you can basically reconstruct the binding pose that this solution corresponds to. So that's enough on the high level. I don't want to dive too deep, but then this problem boils down to finding a clique. So what's a clique? If you have a graph, you can find subgraphs within that larger graph. And this one, for example, would be a subgraph, but it's not a clique because it's not fully connected. There is no edge between these two vertices. And the clique is defined as a fully connected subgraph. So this guy here is a clique because all edges are connected with each other, but it's not the maximal clique because it's not the largest subgraph that is fully connected. That's this guy. And those types of cliques, maximum cliques, is what we're trying to find in this hypergraph that we constructed. And it's a non-trivial problem. It's actually NP hard. And that's why we turned to Xanadu and tried to figure out a way how we can use their quantum hardware. What they're building is a Gaussian boson sampler. That's going to be their first device. It's a special purpose device. You send in photons on one end. There's a large optical array which actually implements a matrix, a unitary K. And then your photons come out on the other end, and you have single photon detectors lined up which measure if a photon comes through or not. And based on that output distribution of photons, you can basically extract subgraphs from your large hypergraph, which is encoded in this matrix. So they basically published on that at some point last year, where they actually found out how you can tweak the parameters to encode any sort of graph in this device. And they also drew some really nice, they had some nice realizations, which means that if you use this device and you sample subgraphs from this Gaussian boson sampler, these subgraphs tend to be very dense, which is great because we're trying to find cliques. and cliques are fully connected, which is maximally dense. So we kind of thought that that might be a good way of sampling cliques. And that's what we tried out. We tried different algorithms. We first started out with the most simple algorithm that you could think of, just random search, sample a subgraph, look at it. Is it a clique? No? Okay, you throw it away, you sample another subgraph. If it's a clique, you store it, and you just keep doing that, hoping that you're going to find large cliques eventually. So we tried that as kind of the baseline that we wanted to compare things to. And then we started getting a little bit more fancy. So these are all classical algorithms that people have used in the traditional world. Here you again sample a subgraph with the Gaussian boson sampler, which might turn out to be this one. The next step is pick the node with the least edges, which is this guy, and remove it. And once you remove that, you determine if your leftover subgraph is a clique. If no, you can repeat, you again pick the least connected node and throw it away, and eventually you're going to converge to a clique, and you can store that and see if it's larger or smaller than what you've already have in memory. And then we said, okay, well, the problem with quantum algorithms papers is often that they don't compare to the state of the art, and we wanted to compare to the state of the art, and the state of the art is dynamic local search, which is like they have won lots of competitions, like MAX-Cliq search competitions. And it's a very complicated algorithm. I'm not going to elaborate it, but we basically just inserted a Gaussian boson sampling step, because usually all these algorithms usually just randomly sample, whereas we have the advantage of using that Gaussian boson sampler, which gives us dense subgraphs rather than just any graph. And so we did that, and we compared it to classical random sampling. So this is greedy shrinking with dynamic local search, and you can see that the success rate of sampling the correct clique increases dramatically when using that Gaussian boson sampling step. All of these things didn't run on actual hardware. That's all like still simulation-based, so also the graphs were still relatively small. But ultimately, if they would have the device, you could scale that up and take a much more complicated representation of your molecule. And it brings me back to this idea of a hybrid pipeline. You extract your pharmacophores on a classical device. You then use this GBS-enhanced algorithm in order to sample the maximum cliques, and then you already have a good idea of the orientation of the molecule, and you can again use restrained docking simulations where you already pull the molecule kind of into that configuration and then just like sample locally around that. So that's the part on molecular docking, and I just wanted to quickly highlight some of the work we are doing on open source software. And that's the exciting part that maybe also some of you, if you're excited about quantum computing, if you're a software developer, but you might have never studied physics, it's something exciting to know that quantum computing is happening, quantum software engineering is kind of becoming a thing, and most of it is actually happening open source. Actually, most of the hardware providers have open source packages, mainly like open source the entire stack, with the exception of maybe some of the compilers. And I always like to make fun of physicists and the way they code, because I myself didn't know how to code before I started this company. And there's a lot of software engineering outstanding. So if you're excited to kind of get your hands dirty and like work with some of these projects, they can even like do sometimes with some help. There's like smaller projects by some grad students that don't know how to like properly package it with pip or things like that. Like there's a lot of things you can do without necessarily having to understand the quantum computing part of it. But it also tends to like be a very nice way of like picking it up, because the documentation sometimes really walks you all the way like nicely from the introduction to quantum computing all the way to like implementing your first algorithms. And so that kind of led, like we were invited to write a review paper that reviewed the entire landscape of open source packages. We evaluated this like quality of documentation, the way they adhere to best practices in the open source space. And we founded this foundation, the Quantum Open Source Foundation, which really aims to, so we have monthly updates on these metrics where we compare projects, and we're trying to kind of get people to answer pull requests faster, improve their documentation, and just be aware of those shortcomings. And we're always happy to have people getting involved. Like you can sign up for the newsletter. We are planning to host a hackathon and like basically kind of issue fixing competitions. We have a conference going on yearly, which is at FOSDEM in Brussels. We have like our own quantum computing stream there, where people come and talk about their projects and kind of try to find contributors that want to join the effort. And that's it from my side. I'm happy to, okay, that's perfect. I'm happy to answer any sort of questions that you have. Thank you.
I'm sure there must be questions. There is one. I'll pass the microphone to you.
Hi, I'm Matthias. Thanks for the talk. I was wondering if you could explain a little bit more about GBS and why it is that you get somehow larger graphs from it. Like, I didn't understand it at all.
Why you get denser graphs from it? Okay, let me just skip back to this. So, I mean, the argument gets involved, but basically the probability of sampling a particular subgraph is linked to this Hafnian, which is a metric. It's kind of like a determinant of a matrix that you can compute. And the Hafnian of that matrix, AS, basically represents that large hypergraph. And the Hafnian for subgraphs of that hypergraph tends to be larger than for less dense graphs. And that's what they basically established here empirically. That was something that they figured out while playing with the device and also kind of like studying the equations. And it might not actually be a great property for some other algorithms where you may, I don't know, you might try to find some sparse subgraphs or something like that. But because of this relation of the Hafnian to the probability of sampling a particular output distribution of this Gaussian boson sampler, that's why that probability tends to be skewed towards dense subgraphs. We can talk about that later in more detail if you want, but it's a kind of involved mathematical argument.
Do I see more questions from the audience? Oh, there's one. Wait, I'm coming around.
Well, transferring that what you said and reversing the direction of thought. If you're applying quantum computing to finding more consistent or improve the performance of finding consistent configurations somehow of your molecules, what does your intuition tell you? Do you think that nature might also have relied on these quantum effects to solve the evolutionary problem to find those configurations? Because there's this riddle that in evolution, so much trial and error must have happened to find these things that there was not enough time for this trial and error. So that now the question arises, might quantum effects also affect larger structures? So I reverse your findings and say, might quantum effects have contributed to constructing these real-life protein configurations?
Frankly, I don't know. But
just asking for your intuition. What does your intuition tell you?
There's actually now a whole field called quantum biology, where they basically look at exactly those things, like quantum effects in macroscopic systems. They, for example, could prove that there's quantum effects that without them, photosynthesis wouldn't be possible. There has obviously been studied, they're trying to, for example, do quantum mechanical folding of proteins, which is extremely difficult to do on supercomputers because it involves too much computation. What they found until this point is that it seems to be fine just using, simulating classical physics to most of the time arrive at the correct configurations. But still, molecular dynamics is not perfect by any means. There's certain types of proteins that you can't fold properly, certain interactions that aren't accounted for. So at the end of the day, yes, if you could simulate the full quantum mechanics of the entire large protein, you would most probably arrive at the correct solution much more likely than trying to use the approximations of classical physics. But to what extent that kind of contributed to evolution, no idea.
I think we had another question on the left-hand side.
When you map your protein structure onto a cubic lattice, how do you estimate or calculate the interaction between two amino acids?
Yeah, that's a good question. Basically, in the papers on arXiv, it's a lot of equations where you're basically trying to derive equations that if two amino acids are neighboring on the lattice, it realizes a certain term that gets added to your energy function. And if they're not, if they're separated by a large distance on the lattice, you do not apply this particular term to your energy function. And you're trying to minimize the energy over that whole thing. So it's basically a lot of sums and products over the entire space of grid points in order to try to figure out, like usually they evaluate to 0, but only in the case when they are next to each other, that term evaluates to 1 and adds that penalty term to your energy function. So that's also how, if you map a problem to the device, you tend to introduce ancillar variables in order to be able to map to the topology. And those ancillar variables, they enter the equation in a similar way. They have a penalty term that if they don't act the same, you apply an energy penalty to make sure that those solutions tend to have very high energy, not popping up in your solution space. So yeah, that's pretty much what it boils down to. When it comes to quantum annealing, you need to really think hard about how you can formulate it in this discrete binary way of a QAOA problem, which is a quadratic unconstrained binary optimization problem. And there's lots of ways. Like people have looked at like aerodynamics, problems in quantum chemistry, and so on. And there's a lot of mappings, kind of a toolbox of elements that you can draw from and apply to your problem.
And there was another question in the first row.
Hi, thank you for your talk. It was really interesting. I was wondering, I recently read that people are having really large breakthroughs with doing protein folding by machine learning or artificial intelligence. Mm-hmm. Do you have any idea, rule of thumb, how the methods, quantum computing for protein folding and machine learning compare in quality or trackable problems?
And the answer is simple. Quantum computing right now doesn't contribute anything meaningful to protein folding, but eventually it will, I think. But to answer that question, like AlphaFold by Google, they had a big breakthrough and they outperformed everyone on that folding competition that happens every 2 years. It's super exciting, and I talked about that yesterday at lunch because it was something that like investors asked us a lot when we raised our seed round right now, like, oh, how are you better than those guys? Like, are they a competition? The thing is that they still used like all of those existing proteins and trained their model on that. Where's that slide? Here. So basically they were smarter than academia in the way that they are very good at software engineering. They applied different ways of mining the existing database. They didn't have any additional proprietary data. They were just better at it, but they still used that data, which means it's very hard to actually come up with completely new scaffolds. So a lot of protein design software that is out there is also based on piecing like existing fragments together. Like you look at your protein, oh, I know how that fragment looks like, I know how that looks like, and they piece it together and that's a model. But usually those models like always stay close to what is known. And that's really kind of what we're trying to surpass, like being able to like show these pharma companies new things that they wouldn't have cooked up in their labs even. So that's kind of like the differentiator that we see there. We don't tend to rely too much on existing training data.
I think we have time for one more question. Whom am I going to pick? Let's go down there.
Hi, you mentioned QAOA, and I've heard that there's a very recent result from the beginning of this year that says if you make your variational quantum circuit deeper, then the gradient becomes exponentially flat. Did you find this to be a problem?
Nope, because we didn't like had large enough problem instances yet. But yeah, like that's a very interesting point actually. It's something that worries me, and I'm now doing a master's right now on the side which kind of focuses on that problem because I want to answer the question. Like those Barron plateaus do occur if you scale up the number of qubits, which is obviously going to kill any sort of quantum advantage if your classical optimizer kind of stumbles in the dark, not being able to find a gradient. But there was also some recent results as a response to that paper where they showed, like the same problem happened in classical neural networks, right? You had vanishing gradients and people came up with like techniques to kind of circumvent that. And they now had some techniques where you can show that if you don't start, like that argument applies to random parameter initialization. If you have a smart guess, a good ansatz, you can basically avoid this. And for certain problem instances, those things don't tend to start. So the question is really to explore that more and to like do analyses of the energy landscapes, trying to develop techniques. Or like, for example, there's an idea of qubit dropout, similar to like a dropout in classical neural networks, where you just measure them halfway through the computation, thereby reducing the space that you're trying to optimize over. So people are very aware of that because it's a big issue when it comes to actually scaling up these hybrid algorithms. Good point.
Thanks.
So finally, thank you for this magnificent talk and for the very interactive Q&A session. Let's give a big hand to Mark. Next up is a very short break where you can switch rooms or just stay here for the next session in this room. Thank you.